a biofuels case study presentation. I'm Ingrid Miller, a product manager for Bio-Rad's Biotechnology Explorer program. I'd like to introduce Cindy Gay, our expert presenter today. Cindy completed both her undergraduate and graduate work in environmental, population, and organismic biology at the University of Colorado. For the past 29 years, she has taught biology to students ranging from middle school to college undergraduates. Cindy is a nationally board certified teacher and is currently teaching biology, AP biology, and anatomy physiology at Steamboat Springs High School in Colorado. She has been recognized for her teaching, curriculum, and staff development as a recipient of the Presidential Awards for Excellence in Science Teaching, as an outstanding biology teacher, the Amgen Award for Excellence in Science Education, and as a Teacher of the Year. Cindy's passion for education really shines through in all she does, which I hope you all see today. If you have questions during today's presentation, you can submit them to the presenters via the text chat. We will try to answer as many questions as possible at the end of the presentation. Just a reminder, this webinar is being recorded and we will send it out to you after the event. Thank you, and I'll hand it over to Cindy now.
Thanks, Ingrid, and thanks to all of you for joining me today to talk about a subject that I love. Proteins and protein enzymes rule the living world and play a key role all year long in my biology and AP biology classes. In this webinar tonight, we'll talk about why it's important to teach enzymes, enzyme function, regulation, and STEM applications, STEM meaning science, technology, engineering, and mathematics. Biofuels as a case study to explore enzyme activity. We'll look at some hands-on labs for teaching enzymes, and again, as Ingrid mentioned, we'll provide time for questions at the end of the webinar. But please remember, as questions arise, please enter them as text chat. So why teach enzymes? Enzymes are clearly present in national and state standards. They feature prominently in the AP and IB exams, and they're an important concept in college classes like general biology, chemistry, and biochemistry. Enzymes spiral throughout a biology curriculum, making appearances all year long, both within a single course as well as throughout a strong secondary and post-secondary education. And best of all, enzymes provide great opportunities to incorporate best teaching practices. There's great stories about enzymes and the substrates they catalyze. There's stories about careers and women in science, like Maud Menton, who we'll meet later in this talk. They help highlight overarching themes like structure and function, unity and diversity, response to stimuli, and homeostasis, all throughout a spiraling curriculum. They provide opportunities to talk about current issues, decision-making, and bioethics. Enzymes can also provide hands-on, highly visual inquiry lab experiences, like AP Lab 2. They provide a context to discuss the nature of science and the importance of an interdisciplinary team approach to problems. So in preparing for this webinar, I did a quick scan through my AP Biology curriculum to see just how often I did mention enzymes throughout the year. And so here's an abbreviated list. Enzymes appeared when we studied biochemistry, looking at protein structure, chemical bond properties, and enzymatic reactions. In cell biology, enzyme regulation, and enzymes appeared in cellular energetic studies and metabolic pathways. When we looked at genetics, enzymes were present looking at the central dogma, in gene regulation, in molecular biology and biotechnology. In physiology, enzyme function plays prominent roles in digestion and neurotransmission, as well as signal transduction and disease. In evolution, thermophilic life forms, such as those whose enzymes became the inspiration for PCR, came up often, as well as mycorrhizal fungi and its relationship with plants and its facilitation of their movement from plants from water to land. In environment and health, we look at biogeochemical cycles. Enzymes play a prominent role in the carbon cycle and the nitrogen cycle, as well as in biofuels. In inquiry, there's all kinds of experimental design and controls for enzymes, PCR, restriction enzymes. So pretty much overall, enzymes are ubiquitous, emphasizing those cross-disciplinary connections and lab techniques. So now let's take a look at why enzymes are so amazing. Living organisms are basically biochemical factories, dependent upon the rapid formation and breaking of chemical bonds. Normally, uncatalyzed reaction rates are slow. Carbon dioxide hydration to carbonic acid occurs roughly at 200 molecules per hour. If we add an enzyme, that rate goes up to 600,000 molecules per second. If you want to increase the likelihood of a chemical reaction, you need to do two things. First, either increase the number of collisions by increasing the temperature or increasing the reactant concentration. Neither one of these is good for living systems because it speeds up all reactions, not just the desired ones. You can also add a catalyst, a substance that enhances the rate of chemical reaction, but's not permanently altered or consumed by the reaction. Now there's two types of biological catalysts: enzymes, which are catalytic proteins, and ribozymes, the subject of Tom Cech's Nobel Prize-winning discovery. Those we're not going to discuss here this evening. Some important properties of enzymes include the fact that they increase the rate of reactions. Increases of 10 to the 7th to 10 to the 9th are not uncommon. Reactions can occur in micro to milliseconds. Enzymes are highly specific, and side products are rarely formed, unlike inorganic catalysts. And finally, regulation is important, and it's possible because of their large and complex structure. In living systems, enzymes are found suspended in the cytoplasm. For example, glucokinase or hexokinase, which is the first step in glycolysis, shown here as a space-filling model and as a ribbon model. Enzymes are also found embedded in cell and organelle membranes as integral proteins. For example, surface receptor proteins, electron transport and ATP synthase enzymes in the mitochondria. In multi-enzyme complexes, we can find enzymes working together. For example, pyruvate dehydrogenase, which is the yellow enzyme here in the picture, it controls entry to the Krebs cycle, is found in a complex with two other enzymes, the red and the green, for a total of 60 subunits. Now, there are several advantages to grouping enzymes into complexes. Since the reaction rate is limited by the frequency of collisions, in a series, the product of one reaction can be delivered as the substrate to the next enzyme. Second, it further decreases the possibility of unwanted side reactions. And finally, all reactions of the complex can then be controlled as a unit. Early classification of enzymes was actually fairly arbitrary. Enzymes were usually named at the whim of the discoverer, and often the name didn't even provide a clue as to its function, for example, catalase. And there were often multiple names for the same enzyme. At the time, enzymes were often named by adding ase to the end of the substrate. For example, sucrase hydrolyzes sucrose. The International Union of Biochemistry instituted a systematic naming scheme. Enzymes are now classified or named according to the type of chemical reaction they catalyze. The name consists of a 4-number classification, a 2-part naming scheme, and a recommended name, often a shorter version of the systematic name. So for example, glucose 6-phosphatase is the 4-number system is 4.1.3.9. Its common name, glucokinase or hexokinase. 6 categories of enzymes are commonly recognized. Oxoreductases transfer electrons in redox reactions. For example, luciferase, which gives fireflies their glow. Transferases transfer functional groups from donor to receptor molecules. For example, hexokinase, step 1 in glycolysis. Hydrolases are our most familiar category. They break bonds by adding water. For example, cellobiase, which is going to be the function or feature prominently in our webinar later. Lyases add or remove groups to make or break double bonds. For example, pyruvate decarboxylase, which is step 1 of 2 in alcohol fermentation. Isomerases cause intermolecular rearrangements into a new isomer. For example, glucose 6-phosphate isomerase, which occurs in step 2 of glycolysis. And finally, ligases create bonds between two substrate molecules. DNA ligase is probably our most familiar example here. But now we get to the really cool part. How do enzymes work? The answer is revealed in the relationship of structure to function. We can look at how enzymes work at two levels, energetically at the level of function, and structurally using fit models. So to start with energetics, catalysis involves increasing the reaction rate by influencing chemical bonds to lower their activation energy. In other words, it provides an alternative pathway that requires less energy. This mechanism of catalysis is threefold. First, it enables the substrate to absorb enough energy to reach the transition state at a moderate temperature. It does this by destabilizing bonds due to physical strain. It also arranges substrates into the correct spatial orientation and causes the chemical change by temporarily adding groups to the substrate and then destabilizing the bonds. So as we look at a typical chemical energetics graph, the transition state is here at the apex of both pathways, and this represents the unstable state of the molecule. The activation energy, shown here, is defined as the amount of energy required to convert 1 mole of reactant, the substrate, from the ground state to the transition state. A misconception alert here that's important to bring up with students is that the standard free energy of the reaction, here, is not altered by catalysts. Further, exergonic reactions are often called spontaneous, but that doesn't mean that they happen instantaneously or even rapidly. So then, the rate or velocity of a biochemical reaction is determined by measuring the change in the concentration of the product or reactant over time. The rate is fast. A single enzyme typically acts on over 1,000 substrate molecules per second, and some even faster. Enzyme kinetics is the quantitative study of enzyme catalysis, and it provides information about reaction rates. It can measure affinities of enzymes for substrates and their inhibitors, and it gives us insight into reaction mechanisms and forces that regulate metabolic pathways. The Michaelis-Menten plot shows the relationship between the maximum velocity that a reaction can attain, Vmax, and the substrate concentration. It also incorporates the Michaelis constant, Km, which is characteristic of the enzyme and substrate under certain conditions. The lower the Michaelis constant, the greater the affinity of the enzyme for the enzyme-substrate complex. This is a great time to introduce history and women in science. In 1903, Victor Henry first introduced the concept of enzyme-substrate complex. In 1913, Leonore Michaelis and Maud Menten developed their equation. Michaelis also discovered the Janus green stain that we commonly use in biochemistry, and he also discovered that keratin, a protein of the hair, is soluble in thioglycolic acid. Thus, the home permanent was born. Maud Menten was a tiny woman with a reputation for erratic driving in her Model T. An avid mountain climber, she went on several expeditions to the Arctic and spoke at least 6 languages. In 1911, she earned one of the first MDs ever awarded to a Canadian woman. However, Canadian universities at the time excluded women from their faculties, so she went to Germany to work with Michaelis. And I'm having trouble getting the slide to appear. Here we go. So now let's look at how enzymes work from a structural perspective. Enzymes are proteins with at least tertiary structure. Again, here's the relationship of structure to function. Their three-dimensional shape is a result of the order of amino acids, the primary structure. The active site, shown here, is a unique, intricately shaped binding surface. It's a small cleft or crevice on a large protein molecule created by 6 to 12 amino acids. And the amino acid side chains then that line the site actively participate in catalytic processes. The active site is used to optimally orient the substrate, and the shape and charge distribution of the active site constrains the motions and the conformations of the substrate, and it forces it to adopt a conformation more like the transition state. The lock and key model, shown here on the left, was first proposed by Emil Fischer in the 1890s to explain enzyme specificity. And what we mean by that is that the active site and the substrate have complementary structures. It's so complementary that the enzyme can actually distinguish between isomers. Sucrase can hydrolyze sucrose, but not maltose or lactose. The substrate's overall shape and charge allow it to enter and interact with the active site. Daniel Koshland's induced fit model improved upon the lock and key model by taking into account the flexible structure of the protein itself. So in this model, the substrate, doesn't actually fit exactly into the rigid active site, but instead non-covalent interactions between the substrate and the enzyme changes the three-dimensional structure of the active site, conforming the active site shape to that of the substrate in its transition state. This example is hexokinase, our step 1 of glycolysis. The substrate is held in the active site by weak interactions, hydrogen bonds, ionic bonds, and hydrophobic interactions. So some key points to emphasize with students is that the active site is a template for substrates to orient them for reaction. The enzyme itself stretches and stresses the substrate to the conformation of the transition state, and this is what lowers the activation energy. Also, the microenvironment that's provided by the active site side chains, for example, decreased pH, facilitates hydrogen ion transfer to the substrate. And that there's a direct participation of the amino acids in that active site. There's a brief covalent bonding of active site side chains and substrates. But the rest of the protein is equally important as well, and it's often overlooked. The rest of the protein provides a framework to align the amino acids of the active site, and it changes shape when it's in an enzyme substrate complex, which is another aspect of the induced fit. It also provides binding sites for enzyme regulation. It's also important to consider the non-protein components in enzyme structure. For example, cofactors are inorganic ions, usually transition metals, magnesium, zinc, copper, that help to orient the substrate. These cofactors could be permanently bound to the enzyme, or they bind loosely and reversibly with the substrate. Coenzymes are large, complex organic molecules. They're changed in the reaction and must often be regenerated. For example, NAD is regenerated from niacin. And most coenzymes are derived from vitamins. Prosthetics are permanently bound molecules that also aid in enzyme function. Now, enzymes in a cell aren't continually catalyzing reactions. Living organisms have a sophisticated mechanism for regulating metabolic biochemical pathways. And this regulation is essential because all living things need to maintain their ordered state. Regulation of pathways results in productions of substances needed for cell structure and function just when they're needed and without wasting cell resources. This also helps cells conserve energy and resources. Cells consume just enough nutrients to meet the energy requirements by constantly controlling energy-generating pathways. It also allows them to respond rapidly to environmental changes. Cells have to make quick adjustments to changes in pH, to temperature, ionic strength, and nutrient concentrations by changing their pathway reaction rates. So now the question is, how do they do that? They can do it at the level of organismal regulation. They can do it at the level of genetic control, which occurs prior to enzyme production. So this would include enzyme induction, which is the synthesis of enzymes in response to changing metabolic needs. For example, the lac and trip operon in E. coli, as well as signal transduction pathways. It can be regulated by covalent modification. Now this occurs post-enzyme production. And these covalent modifications in enzyme structure allow reversible interconversion between the active and inactive forms of the enzyme. For example, chymotrypsin in the pancreas is activated when it's secreted into the small intestine. Here's a diagram of an enzyme in its typical active form. Inhibition can also help regulate many pathways with naturally occurring inhibitors. Artificial inhibitors are used to treat disease, kill pests, and study how enzymes work. Inhibitors can be either reversible or irreversible. Irreversible involves covalent binding of the inhibitor to side chains at the active site, and that permanently inactivates the enzyme, shown here in the lower left. For example, DIPF is a nerve gas. It permanently inhibits acetylcholinesterase. It was used in the 1995 sarin attack in a Tokyo subway. Malathion pesticide is a DIPF derivative, and it inhibits insect acetylcholinesterase, but not mammalian. Now, reversible inhibition, the inhibitory effect can be counteracted by increasing substrate concentration or removing the inhibitory compound. Inhibition can also be either competitive or non-competitive. In competitive inhibition, the inhibitor mimics the substrate. It's similar enough to bind non-covalently in the active site, but doesn't actually cause a reaction. Non-competitive binds at a site distinct from the active site, causing a change in shape of the enzyme that alters its reactivity, shown here in the lower right. Allosteric inhibition occurs when an effector, either an activator or an inhibitor molecule, binds to a site other than the active site. It also induces the enzyme to change its shape between the active and inactive forms. Finally, enzyme regulation can also be achieved by compartmentalization. This compartmentalization is created by the cellular infrastructure, and it facilitates control by separating competing reactions and creating diffusion barriers. It keeps the concentration of the substrate and the enzyme high, which is important for those multi-enzyme complexes. It also provides damage control by isolating potentially toxic reaction products. The rate of an enzymatic reaction can also be affected by environmental conditions, such as temperature. In general, increased temperature also increases the rate of the reaction due to an increased number of collisions. Therefore, the reaction velocity increases as more molecules have enough energy to enter the transition state. However, enzymes can become denatured at high temperatures. The high thermal agitation disrupts the hydrogen bonds, the ionic bonds, and other weak interactions. So as a result, enzymes have an optimal temperature, the temperature at which the enzyme operates at maximum efficiency. In humans, that temperature is somewhere between 35 and 40 degrees C for most of our enzymes. However, for hot springs bacteria, the optimal temperature is 70 degrees or higher. We use these enzymes in PCR because they can withstand the high temperatures of the process. pH, or hydrogen ion concentration, affects enzymes in several ways. So remember that catalytic activity is related to the ionic state of the active site. Changing the pH affects the ionization of the active site. The substrates can also be affected, altering their ability to bind with the active site. These changes in the ionizable groups can change the whole three-dimensional structure of the enzyme, thus denaturing it. Now, there are a few enzymes that can tolerate large changes in pH, but most are active only within a very narrow pH range, which underscores the importance of buffers. There's also, just like temperature, an optimum pH. The pH value for the maximum enzyme activity for pepsin, found in the stomach, is about 2, while chymotrypsin, found in the small intestine, is about 8. Salinity works similarly to pH by changing the charge of the side chains in the active site. Substrate and enzyme concentration changes can both increase the initial rate of reaction, but they don't influence the final equilibrium. Substrate concentration determines just how fast the enzyme can find and bind to the substrate. Increasing the substrate concentration increases the frequency of finding the active site until all the active sites are full or saturated, and so therefore the initial rate is higher. If we increase the enzyme concentration, similarly, we're going to cause the initial rate to be higher. However, with enough time, the same amount of product is formed, which is limited by the amount of the substrate. So at this point, I'd like to do a process check. So far, we've talked about the background of enzymes, their function, their regulation. And before we go on to talk about the applications of enzyme, I want to remind you to send your questions via text chat. And I also want to remind you that this presentation will be available to all participants. It's being recorded, and we'll send out an email with a link to this recording and copies of the slides. So now let's talk about applications of enzymes. Many processes important to health, technology, and society involve enzymes and their regulation. And these real-world case studies provide relevant context to the study of enzymes and provide even more great stories for your lessons. Diagnostic medicine uses enzymes such as LDH, lactate dehydrogenase, that leak from damaged heart tissue in the detection of heart attacks. In medical treatment, antibiotics use inhibition to decrease or completely eliminate the activity of specific enzymes used in bacterial cell wall formation. The most effective AIDS treatment is a multi-drug therapy that uses protease inhibitors to prevent formation of new viruses. In pharmacogenetics, we use restriction enzymes in the creation of plasmid vectors in bacterial transformation to produce human insulin. In industrial applications, it's important to inhibit enzyme activity in food preservation and in developing effective poisons, such as the malathion example we discussed earlier. Consumer products, such as laundry detergents and cleaning products, accounts for about 1/4 of all commercial enzyme applications. And biofuel production, enzymes play a critical role in our ability to produce ethanol from plant materials. So now we'll shift our focus to the role of enzymes in biofuel production. Now, biofuels are an awesome case study for understanding enzymes and factors that affect enzymatic reactions, because enzymes play a central role in the production of biofuels. The study of biofuels itself creates connections between central concepts in both biology and environmental science. There's currently great interest in renewable energy, both from an environmental perspective as well as reducing our dependence on foreign oil. Biofuels as a case study provides many opportunities to incorporate STEM applications in the curriculum, and perhaps most important to me, it's an important topic to consider if we want to create educated global citizens. My students showed more interest when the study of enzymes was linked to biofuels because of the timeliness and relevance of biofuels in the media. Even though we focused on enzymes and biofuels in the fall, as we were reviewing C4 plants this spring, one student suggested that they would make better candidates for biofuel production than C3 plants, demonstrating an understanding in both subjects, and I was so excited. Biomass energy, or bioenergy, is energy from plants and plant-derived material. Wood is the largest source of biomass energy used today, but other sources include food crops, grassy and woody plants, forestry and agricultural byproducts, oil-rich algae, and organic components of municipal and industrial waste. There's many benefits to using biomass energy. Perhaps the greatest is the potential to reduce greenhouse emissions, because using biomass energy is carbon neutral. So if we want to understand global warming, we need to be able to discriminate between the long-term and short-term carbon cycles. Now, burning fossil fuels increases the concentration of carbon dioxide in the atmosphere, creating the greenhouse effect. This carbon was trapped billions of years ago and is not part of the short-term carbon cycle. On the other hand, burning biofuels doesn't contribute to global warming and is considered to be carbon neutral since the carbon was recently removed as part of the short-term cycle. However, we have to also consider the fossil fuel used to grow and process the biomass. Fossil fuel in fertilizer and used to run farm equipment, but it still results in lower greenhouse gas production. It takes about 1 gallon of gas to produce 5 gallons of ethanol. The net greenhouse gas reduction from using corn grain is about 20%, from soybeans 80%. Ethanol from corn stover, between the reduction of gas use and the electricity we get from burning the lignin, reduces about 113%. And I'd like to publicly thank the Great Lakes Biofuel Research Center for sharing their images for this presentation. They have a wealth of background material as well as some really great teacher and student resources. And I'll provide their contact information at the end of the webinar so that you can take advantage of their extensive resources. So as we continue to look at the benefits of using biomass energy, as we said in the last slide, it was carbon neutral. It also helps support U.S. agricultural and forest industries, as well as reduce our dependence on foreign oil. Petroleum, diesel, and gasoline consist of literally hundreds of different hydrocarbons, including many that are toxic: benzene, toluene, xylene. They produce carbon monoxide, nitrous and sulfur oxides, and particulates. Biofuels can reduce these emissions and are used as fuel oxygenates, and they increase the completion of combustion, which reduces carbon monoxide. Vehicles can actually run on 100% ethanol. For example, all the city buses in Brazil run on pure ethanol. Many cars can also run on E85, which is about 85% ethanol. For example, the Ford Explorer, the Toyota Tundra, and the Saab 9-3. Bioethanol is alcohol made from fermenting sugar. So here's a great curricular tie to anaerobic cellular respiration. Bioethanol can be used to fuel cars in its pure form, but it's usually used as a gasoline additive to increase octane and improve emissions. From their studies of cellular respiration, students should be able to recall that the starting molecule of fermentation is glucose. Sources of glucose in plants are sugar, starches, and cellulose. In the first generation of biofuels, the glucose source for ethanol production came from sugar, primarily from sugarcane and beets, or from starch crops, grains including corn, primarily food crops. Now, ethanol production from sugar is about as simple as it gets, using fermentation that we've learned about in cell respiration. A second step, though, using heat or enzymes, is needed to hydrolyze starch and sugars into glucose, which can then undergo fermentation to ethanol. So here are some more great curricular ties both to macromolecules and hydrolytic enzymes like amylase, which break down those alpha linkages between glucose molecules. Amylase and starch labs demonstrating this process have been a long-term staple in most biology classrooms. In the U.S., corn has been the primary glucose source in bioethanol production. However, there are many concerns with the use of corn, including the fact that it's an annual crop. It's heavily dependent on chemical inputs. So here's another curricular tie to biogeochemical cycles. Crops that are harvested for biofuels remove nitrogen from the soil, and inorganic fertilizers that put it back are made from fossil fuels. Nitrogen-based greenhouse gases are up to 300 times more potent than carbon-based greenhouse gases. Tilling and harvesting increases soil erosion. This map shows the Mississippi River Basin and the resulting hypoxic or dead zone in the Gulf of Mexico. Finally, annual crops provide little below-ground carbon storage. There's also the issue of food versus fuel, and this is a huge concern, as is the reduction of wildlife habitat and the impacts on biodiversity. Second-generation biofuels are produced from cellulose rather than starch and sugar. We know that all plant cell walls are made primarily of cellulose. There are several components of plant cell walls that we need to consider here. Cellulose, shown in yellow, is the most common form of carbon in biomass. It makes up about 40 to 60% of the biomass by weight. It's a polysaccharide of glucose, but its crystalline structure makes it resistant to hydrolysis. Hemicellulose, shown here in blue, makes up about 20 to 40% of the biomass by weight. It's made from a variety of 5 and 6 carbon sugars, and while it's easy to hydrolyze it down to simple sugars, it's really difficult to ferment to alcohol. Finally, lignin, shown here in orange, is about anywhere from 10 to 24% biomass weight, And it provides the structural integrity in plants. It's a highly complex aromatic molecule found in secondary plant cell walls. And it remains as residual matter after ethanol conversion, but it still contains a lot of free energy, and we can burn it to produce steam and electricity for the actual biomass ethanol process. There are, however, some hurdles in using cellulose as the glucose source for bioethanol production. Cell wall composition varies, and we currently lack an efficient way to break down cellulose and convert it to glucose. And here's where enzymes come in. So the question is, can we find an efficient way to produce glucose from cellulose? The answer lies with enzymes. And to begin to think about this question, we need to understand the main steps of cellulosic biofuel production. A quick overview of this process shows three main steps. There's pretreatment, shown here in the blue on the left, hydrolysis that uses cellulase and fermentation, this is shown in green here in the center, and then product recovery, shown on the right, also in blue. With respect to our study of enzymes, as well as the focus of current biofuel research, today we'll limit our discussion to just the first two steps of this process. So the pretreatment step first requires size reduction. We need to do that for easier handling and to increase the processing efficiency. So agricultural residues are ground up and forest products are chipped. The actual pretreatment requires breaking down the cell wall and separating the lignin, hemicelluloses, and the cellulose components. The hemicellulose fraction is then broken down into simple sugars like xylose, arabinose, and galactose using heat, acid, and enzymes. This pretreatment process is analogous to mechanical digestion in the mouth, while the hydrolysis and fermentation processes are analogous to chemical digestion. Another great connection at this point is with the arabinose sugar produced from the hemicellulose. So recall that arabinose is used in the operon system in the pea-glow bacterial transformation. Hydrolysis is a chemical reaction that converts complex polymers to monomers. In this case, we're going to convert polysaccharides to simple sugars. Here's another great opportunity to underscore the importance of structure in biological processes. Humans can digest starch, but not cellulose, even though they're both polymers of glucose. So why is that? Well, starch is a polymer of alpha-glucose monomers, while cellulose is a polymer of beta-glucose monomers. The difference is easily seen if you follow the oxygens in the carbon rings. So here we follow the oxygens in the starch, in the alpha-glucose, and down below, if we follow the oxygens, we see a very different pattern. This simple difference means that we have to require different enzymes for hydrolysis. Recall that hydrolysis of cellulose is a really difficult step because of its crystalline structure. Cellulose molecules are attracted to each other by hydrogen bonds, creating microfibrils made of 60 to 80 individual strands of cellulose. We can hydrolyze cellulose to glucose using a variety of beta-glucosidase enzymes. The one that we'll be interested in today is cellobiase. It's also known as beta-glucosidase. It breaks the disaccharide cellobiose into two glucose molecules. The goal to develop enzymes that make this step more efficient and economic is very important. One important issue is finding a good source for this enzyme. And if we take one more look at cellulose and its crystalline structure, we can see that it requires multiple different enzymes to break it completely down to glucose. One group of enzymes are the exocellulases, shown in pink. They break the covalent bonds at the ends of the cellulose molecule. Another group are the endocellulases, shown in red, that break the internal bonds of cellulose here. And finally, the final group are the cellobiases, shown in green. They break the dimer cellobiose into individual glucose molecules. This enzyme is going to be very important in our upcoming lab. So, to find a good source for the various cellulase enzymes, we need to go bioprospecting. If we can find and study organisms that produce this enzyme, we can make the biofuel process more efficient and more economical. So, some additional considerations that we have to include are the issue that the industrial hydrolysis often requires high heat or extreme pH, which denatures many enzymes. So, how do we find and engineer enzymes to withstand these conditions? We can look for enzymes, bacteria or fungi that live in similar environments, hot springs, acidic bogs, or alkaline soda lakes. We can mutate the microbes with UV or mutagenic chemicals to produce the desired enzymes and characteristics. Once we've found the genes, we can use genetic engineering to clone the gene that codes for the cellulase enzyme of interest. We can copy the gene using error-prone PCR, producing an assortment of mutations that are then put in plasmids. We insert those into bacteria, and then the bacteria are screened for cellulase activity in our desired conditions. Now, there's a myriad of labs for teaching about enzymes. For the past 20 years, I've used the traditional AP Lab 2, the hydrogen peroxide and catalase titration lab, to test enzyme reaction rates in my AP Bio classes. This year, I was fortunate to be able to beta test the Bio-Rad biofuel enzyme kit with my students. The kit aligned perfectly with Lab 2, and it actually allowed more thorough coverage of the topic than the traditional catalase titration lab. Rather than an indirect measurement of the substrate remaining, students actually directly measured the time course and the products of an enzymatic reaction. They also measured effects of temperature, pH, substrate, and enzyme concentrations on enzyme activity. In the context of bioprospecting for cellulase enzymes, students also conduct an inquiry investigation comparing the enzymatic properties of mushroom extracts. All of the experiments can be run either qualitatively or quantitatively, and the data is robust with either collection method. You can use the data to construct a standard curve, and you can do a Michaelis-Menten analysis if you desire. In each kit, there's enough material for 8 student workstations, 4 students per workstation, to run all 6 activities. So the kit supports 6 activities. The first experiment serves as a control for the rest of the labs and determines the rate of reaction in the presence or absence of the enzyme. Activities 2 and 3 measure the environmental effects of temperature and pH on enzymes. Activities 4 and 5 examine the effects of enzyme and substrate concentration on reaction rate. And Activity 6 allows students to test the ability of mushroom extracts to catalyze the breakdown of substrate, which in all 6 of these experiments is cellobiose. So recall from our earlier discussion that cellobiose is a dimer that's hydrolyzed between the 1,4 linkage into cellobiase, producing 2 glucose molecules. Unfortunately, though, cellobiose and glucose are both colorless when dissolved, which makes data collection difficult. So, if we remove one of the glucose molecules and replace it with p-nitrophenol, we create the artificial substrate p-nitrophenol glucopyranoside. This molecule will allow us to track the progress of the reaction by monitoring the appearance of a yellow color. Not to mention, my students just loved saying p-nitrophenol glucopyranoside. So, when p-nitrophenol glucopyranoside is hydrolyzed by cellobiase, it breaks into a glucose monomer and p-nitrophenol. In basic conditions, the p-nitrophenol turns yellow. The darker the yellow color, the more product's been formed. So recall that the rate of reaction can be measured by measuring the change in product concentration over time. Now, qualitatively, this can be measured by comparing the final color of the reaction with a set of known standards. Quantitatively, we can use a spectrophotometer or a colorimeter to measure the change in absorbance, and then we construct a standard curve to determine the change in product concentration. The protocol is simple and straightforward. Substrate samples are prepared according to the variable being tested. The cuvettes are filled with a basic stop solution. The stop solution serves to both denature the enzyme, stopping the reaction, and create the basic environment needed to turn the p-nitrophenol yellow. An enzyme is added to the substrate and, at the determined time, a sample of the reaction is added to the stop solution. Since the reacting solution is added to the stop solution, you can use a single reaction tube and remove samples periodically at the designated times. The completed reactions then can be analyzed either qualitatively or quantitatively. Because the protocol is a direct measure of enzyme reactivity, as well as a connection to a relevant issue, the series of experiments becomes a powerful tool for understanding enzymes and biofuels, as well as reinforcing key concepts central to biology. As an added bonus, you can download the entire curriculum for this kit online at the Bio-Rad website. So this fall, I used the Biofuels enzyme kit with my 2 AP biology classes in place of the traditional Lab 2, the traditional catalase peroxide titration. I teach in a 4x4 block schedule with 95-minute classes meeting every other day. So prior to the lab, we had discussions around enzyme structure, function, and regulation, just like normal, and added an introduction to biofuels. The first lab period, all groups, and they were in groups of 3 to 4 students per groups, did activity 1 to ground in the protocol and establish their controls. Then I divided the class in half. Half of the group measured the effects of temperature, activity 2. The other half measured the effects of pH, activity 3. I had my students measure these reaction rates both qualitatively and quantitatively. On the first day, they were able to complete all the experiments and compare their results, and we had a great talk about the significance of their data. On the second day, I again divided the class into 2. Half the class measured the effects of enzyme concentration, activity 4. The other half measured the effects of substrate concentration, activity 5. They then all completed activity 6, testing the mushroom extracts. And many groups had time to go back and complete the activities that they had not done on day 1 or 2, as well as check results that they felt uncertain of. So perhaps my biggest aha from using this protocol was that students were really able to focus on the content rather than the protocol itself. Because the protocol was so simple, I heard students discussing enzyme properties and the effects of variables on reaction rates, rather than asking each other, hey, what do I do next? They were able to complete the labs in a timely manner, and they redid activities that they felt did not produce reasonable data. And because they were focusing on content, they were able to discriminate what was reasonable data and what wasn't. The data generated rich discussion among the students, not only about enzymes and the rates of catalyzed reactions, but also about biofuel production and industrial applications of science. Throughout the course of the year, the ideas and the concepts that were introduced by these lab activities have reemerged. Not only was it a highly visual experience that resulted in really deep understanding of enzymatic reactions, but with the increasing frequency of both enzymes and biofuels in both the class and the popular media, it's continued to resurface the conversations that we started in the fall. And so now at this time, I'm going to turn the presentation back to Ingrid, who will share some resources as well as a special offer with you.
Thank you, Cindy. Before we answer questions, I'd like to let you know that there are several resources available to you regarding enzymes and biofuels. These will be sent along to you with the playback of the webinar in a few days. Also, we'd like to extend a special offer to each of you. When you purchase the new Biofuel Enzyme Kit, you can receive a free P1000 Classroom Digital Micropipette over a $100 value. Please contact us at biotechnology_explorer@bio-rad.com to receive your quote for this special offer. Also, at the end of this session, you will have the opportunity to complete the exit survey to give us feedback on this session and share other topics you'd like to have us present. Now, I'd like to open the floor to questions. And I'll read the questions as they come in. And to the text chat, and Cindy will answer them. So the first question I have is, biofuels from corn, let's say, reduces the available corn for animal feeds, thereby driving those costs up. Wouldn't algae be a better source?
And so the answer to that is perhaps, although there's definitely some issues. Right now, we know how to produce bioethanol from the corn kernels, which is the issue of food. Many research centers are still working on how to actually efficiently and economically extract ethanol from cellulose by breaking it down. And also, the algae itself requires a lot of water to grow, as well as being bioengineered, and there's a lot of concern out there with GMO as well.
The next question is, can I share your PowerPoint with other instructors or not? And I can go ahead and answer that one, Cindy, if you don't mind. Yes, please do share the PowerPoint. And they're also editable, so you can take content and use it for your own presentations in the future. Okay, and the next question is, are there any safety issues with the kit, and how about disposal of any chemicals? And I'm going to pass that question to Michelle Gilbert, the R&D scientist who developed the kit and understands more than we know.
Hi, it's an excellent question. We can't tell you specifically for every state and location on how to dispose of chemicals because that is state specific. But some basics are that we don't have anything below pH 5 or above pH 9.5. So no super strong acids or super strong bases. And the paranitrophenol is at a low enough level that is not considered hazardous, but again, you do want to check with your school system or your regulatory.
Okay, thank you, Michelle. The next question is, my school has 55-minute class periods. Are there adjustments to the protocol that allow for appropriate stopping spots for this kind of schedule?
Ooh. Absolutely. Each activity, if you look in the curriculum guide, is estimated at about 45 minutes. And I would say that when my students all collectively did activity 1, which was establishing the baseline, it took them about 40 minutes or so. And then once they were feeling comfortable with the curriculum, they probably completed the other activities anywhere from 20 to 35, 40 minutes, depending upon the proficiency of the lab group. So you could easily break this up into 40, 45-minute sections. You could also, if you don't want to take 6 40-minute class periods, you could also divide the class up and have part of the class doing one activity, for example, temperature, and another group doing pH, and then compare and pool data that way.
Next question. You did not explain how the mushroom extracts were used in this exercise specifically. Could you elaborate a bit on this? Do you provide the mushroom extracts, or do we go shopping and provide our own?
Oh, that's a great question. So the mushroom extracts are not provided. You should buy your mushrooms sources from a grocery store rather than going out and collecting your own mushrooms because there's lots of pretty poisonous mushrooms out there. When I did this lab with my students, I live in a small mountain town, so we don't have a wide assortment of mushrooms available. And so we used button mushrooms and criminy mushrooms. And one student in a pinch actually brought in canned mushrooms, which actually elicited some really interesting discussions. The canned mushrooms had no enzyme activity. And the students were able to sort of figure out that that was probably due to the fact that the enzymes were denatured from the heat of the canning process.
The next question is, how much time does it take 8 lab groups to complete the labs and read their results in the spectrophotometer? I have 50-minute lab periods.
And so It would be definitely possible for those 8 groups to do that. You could set it up in some sort of organized system. Once the cuvettes are stopped, the reaction has stopped, and the cuvettes can hold for an indefinite period of time. And you can also have them compare their results qualitatively, because you also prepare a set of standards that students can compare the color to. And either qualitatively or quantitatively, I had my students do both, and we got very, very comparable data using both methods.
The next question is, does the indicator in the kit work for any other enzymes? And I'll let Michelle answer this one.
The substrate in the kit would work for any enzyme that's what's called a beta glucosidase enzyme. It's not you can find those in bitter almonds. Some other health supplements claim they have them. We haven't tested it with those, but it'd be a fun experiment for students to test.
The next question is, can you use dry mushrooms, or do you get the same result as with the canned?
I have not used dry mushrooms, but I know that Michelle Gilbert, the developer of the lab, used dry shiitake mushrooms, I believe, and got great results from them.
That is correct. The next question is, going back to the issue on food versus biofuel with corn, what are the problems with using the unedible part of the corn, for example, the husk?
At that point, we're not looking at breaking down starch anymore. We're back to breaking down cellulose and breaking down the plant cell walls. And this is the step that's really tricky in terms of finding the right enzymes to break it down in an efficient and economical way. And that would also be true then for the corn stover, which is the leftover stalk of the corn. And so if we can find an efficient way to do that, then we could still use the corn kernels for a food source, either for humans or livestock, and then use the corn stover for biofuel production.
Thank you, Cindy. The next question is alluding to, I believe, wondering what your opinion is on the applicability of this topic or this combination of topics for different biology type classes, higher level, low level, or versus anatomy and physiology type classes.
I would find both these topics and the labs, they fit really well into my AP biology class, but they would also fit equally well into my 10th grade biology class. My class in my school, biology is a 10th grade level class, and it's full inclusion. So I could have the valedictorian sitting next to a severe needs student. And what I find is that these current topics really help students both at the low end as well as the high end put their hands around some of these more abstract concepts like enzyme kinetics. Depending on the context, I think you could use it in a wide variety of life science classes.
The next question is, if there are less than 8 groups, will the reagents keep until next year? And I'm going to let Michelle address this one.
The buffers will keep, not a problem there, and the substrate as well, or the standards and the enzyme in the concentrated forms that we give them to you in the lab. The trick is you would need a microbalance of some form. We give you the substrate in powder form, and you'd need to weigh out part of it to reconstitute for the first lab you're using it and the rest for a later lab, because once it's been reconstituted, then it has about a 1 month shelf life or so.
Thank you, Michelle. And one more question on the use of corn as a biofuel. In using the corn as a source of biofuel, how much of the corn is used and what is left over that is a disposal issue?
So presently, right now, it's primarily the kernels that are used, and then the rest of the stock we're not able to use very well because, again, it's that struggle with breaking down the cellulose. Ideally, if we can if we find efficient ways and enzymes to break down that cellulose, then we can pretty much use all of the plant. And the lignin that we can't break down, that really that provides the structural integrity, actually still has a lot of high energy, and that can be burned